Hello everyone, this is Matthew Wakeling. This is just a quick video to show how whole genome sequencing works, short read sequencing, also known as next generation sequencing. I'm just using this picture because it's colourful and whole genomes are kind of long and thin, so I've just made it long and thin. But we also have two copies of our genome in each cell, so we've made two copies. And we also have variants. These are what we're looking for when we're doing sequencing. So I'm going to add these coloured lines just to show that there are variants. Now when we receive a DNA sample, we have lots of copies of these genomes in the sample, and so got lots of copies. And one of the first things we do is we fragment that DNA into lots of smaller pieces of DNA, and we end up with these small fragments of DNA being mixed up in a tube together. And some of these fragments are too big and some of them are too small to be sequenced on the short read sequencing machine. So we have a size selection step that gets rid of all of the ones that are too big or too small. So now we have a test tube full of DNA fragments and we don't know what they are. So I'm just going to grey them out to show that this is DNA that is of different lengths. We don't know what they are. The sequencing machine will sequence from the left and then it'll sequence from the right. And this is why it's called paired end sequencing, because you've got two reads that are paired together from the same fragment. And this gap in the middle, we don't know how big it is, so I'm just going to have a line showing that. So once we've done this, we've got data in the computer, and we can then process it by aligning the reads to the genome, the human reference genome. And that's by just taking each fragment one by one and finding the part of the genome reference that it matches the closest. And then once we've done that, we get this big pile-up of all of the sequencing reads that we've made. And this is great. We can see where there are places where it's different to the human reference, but we're actually not interested in the places where it's the same as the human reference. We just fade that away, and now you can very clearly see where we have the variants found by the sequencing.